Hi everyone, thanks so much for joining today. My name's Jan Strugnell and I'm the Director for the Centre for Sustainable Tropical Fisheries and Aquaculture, or the CSTFA. I'd like to welcome you to our seminar series. The CSTFA is based at James Cook Uni in Townsville, for those of you that don't know and have joined us through Twitter. And we're a research provider for sustainable tropical fisheries and aquaculture research globally. And we provide world-class, multidisciplinary, solutions-focused research for Australian and international resource managers, both in government and the private sector. And today, it's my really great pleasure to welcome Abbi Scott. So Abbi is a PhD candidate in the TropWater Seagrass ecology team at JCU. And her research uses field-based experiments to understand the impact of herbivory in the Great Barrier Reef seagrass meadows. Abbi also works with the TropWater team on long-term seagrass monitoring projects throughout Queensland and has a background in working in the UK on rocky shore ecology, citizen science and seagrass mapping projects. If you have any questions, I'd ask you to please put them in the Q&A at the which you'll find at the bottom of your screen, or if you just let me know you have a question there at the end of the webinar, we'll arrange for you to ask your question to Abbi. So thanks very much, Abbi. We really appreciate your time and speaking to us.
Great, thank you very much for having me, and thanks for everyone for coming along to hear about some of my PhD research. So as you can see from my title and my title slide, I'm looking at the impacts of herbivores as ecosystem engineers in tropical seagrass meadows. So I'm going to talk you through some of my PhD work and some of the work that's been done as part of the project that my PhD is one part of. So I'm hoping that a lot of you are familiar with seagrasses within the Great Barrier Reef on our doorstep, but just in case you're not, I'll give you a quick overview of them. So seagrasses are the only flowering plant we have in the marine environment, and they can be found across the globe. So from Australia up to England, where I used to work on seagrasses, everywhere apart from the poles. So we've got 72 species of seagrass around the world. And we're very lucky in the Great Barrier Reef to be in the most diverse bioregion for seagrasses, the tropical Indo-Pacific. So within the Great Barrier Reef, we've got between 12 and 15 species of seagrass, depending on which taxonomist you ask. So they're really, really diverse, and they can be found in the GBR down to a depth of 60 meters because the water quality is so good and light can get down that deep. So like plants, they need light to photosynthesize. And they're really important in terms of their linkages with other habitats. So mangroves and coral reefs, and there's lots of species that are moving between these habitats and using all three of them. And they cover a really large area. So we actually have a larger area of seagrass in the Great Barrier Reef than we do of coral. So maybe we should call it the Great Barrier Seagrass Meadow. That hasn't quite caught on yet, but I'm sure it's only a matter of time. And they're a really important food source for a range of herbivores. So that's what I'm going to talk to you about today. That's where my research is focused. And they provide a range of ecosystem services that benefit wildlife, and they benefit us as well. So for example, they are a biodiversity hotspot. So they provide a structured habitat, and we can find over 30 times more species within a seagrass meadow than adjacent bare sediment. And there's also over 700 species of fish alone that use the seagrass meadows within the tropical Indo-Pacific. They're really good at storing carbon, so up to 35 times more effective than rainforest in taking up carbon. And they can keep this stored in their sediments so that it's locked away for millennia. They're a really productive ecosystem, so they can have really fast growth rates in the tropics, and they're able to take up nutrients from the water column as they're growing. And they can trap sediment. So by having a structure with the blades, they can trap sediment, which helps to keep the water column clear and also increases the height of the seabed and protects coastlines from flooding. They're really important in terms of their fisheries value as well. So providing a home for juvenile fish and a foraging ground. And they're also a home for lots of invertebrates and that people will go and glean from the meadows at low tide often. So they support up to 20% of the world's biggest fisheries. And they're also really important in terms of the trophic transfers to other habitats. So they'll export particulate organic matter and detritus to other habitats, and then animals who are moving between these habitats and feeding, there'll be an export of the animals themselves and moving with them nutrients and other things. So they're really important in terms of their linkages on the GBR. So at the JCU Trop WATER Seagrass Ecology Lab, we're based up in Cairns, and we do a lot of research in terms of monitoring seagrass meadows throughout Queensland, also looking at their fisheries values, value to herbivores, looking at light levels, and really trying to apply this research to management measures. So we're monitoring seagrasses throughout the coast of Queensland, and our methods of monitoring have evolved over the years, from using dirt bikes and hovercrafts on mudflats to now we use helicopters, drop-down cameras, and we'll also go out at low tide and monitor the meadows and snorkel. And so we've got monitoring programs running throughout Queensland, and some of these have been going for 25 years continuously. So we've got a really good idea of how the seagrasses are looking, how they're changing, and the kind of measures you need to put in place to make sure that these seagrass meadows stay healthy. So as I mentioned, my research is looking at herbivory in seagrass meadows. So that's what I'm going to focus on for the rest of this talk. And I'll talk you through some of the PhD work that I've done looking at herbivory in seagrasses, not just by these kind of green turtles that you can see hopefully in this video, but by all of the herbivores. So why are we interested in the fate of seagrass productivity in the Great Barrier Reef? Well, if you go down to South Australia, you'll see these kind of scenes with large amounts of dead seagrass wrack washed up on the shore. And you can see that there's a big pile there with one relatively small seagrass scientist for scale. Whereas if you come to Queensland and come up to Green Island or any of the beaches around here, you won't see as much seagrass washed up, even though we know that we have really large areas of productive seagrass. So the question that we're asking is, where is all this seagrass productivity going? And the answer that we've come up with, or what we want to investigate, is the role of herbivory. And the role of herbivory on land has been well understood for a long time now. So since the 80s, we've had big studies looking at herbivory in grasslands on land, and lots of different exclusion studies throughout the world. And more recent technology is allowing us to get a bigger understanding of herbivory on land on a more bite-by-bite basis. And so really getting an in-depth understanding. Whereas in seagrass meadows, for a long time, it was thought that there wasn't much grazing. Seagrasses were not palatable and were not really being consumed at all. It's only in more recent years that we've started to understand that actually seagrasses are a really important food source, and rates of herbivory within seagrass meadows can be very high. And so we've started to see studies looking at, for example here, turtle herbivory throughout the world, and finding that in areas where there are large populations of turtles, their herbivory can be really significant. And also in terms of the food web as a whole, the influence of predators and top-down controls on those herbivores and what that means for the seagrass meadow. And we're getting a more in-depth understanding of the different groups of herbivores as well. So we saw there the megaherbivores, but fish can also have a really important impact on seagrass meadows. And this is the result when you exclude them from areas in the med. And sea urchins as well. So they can actually overgraze seagrass meadows and cause large-scale declines. And then we're finding out more about the smaller invertebrates that are also grazing within the meadows and what their impact is. So we know that herbivory in seagrasses is really important. We know that in the Great Barrier Reef, we have large areas of seagrass, large populations of herbivores, but we don't really know much about how herbivory is impacting seagrass meadows in the Great Barrier Reef. So this is what I'm looking into and the ARC project that I'm a part of. So who are these herbivores in the Great Barrier Reef? Well, first up, we've got the megaherbivores, the largest ones, turtles and green turtles and dugong. So seagrass is their main food source, and they can consume large amounts of seagrass, particularly the dugong, who focus on seagrass and are mainly consuming seagrass, only consuming other things incidentally, whereas green turtles can switch up their diet a little bit more. Then the macro herbivores, so the fish, particularly rabbitfish and parrotfish and urchins that will live and feed in the seagrass meadow. And lastly, the mesoherbivores, so the small invertebrates that live on the seagrass blades and are primarily feeding on the epiphytes. And we know that these are really important in temperate seagrass meadows in terms of controlling epiphyte growth on the seagrass blades, but we don't really have as much of an idea how important they are in the tropics and particularly within the Great Barrier Reef. So the aims of my PhD as a whole are to understand how herbivore groups interact to structure meadows, identify the important herbivores within the Great Barrier Reef seagrass meadows, and try and understand how herbivory might modify ecosystem service delivery in seagrass meadows. So what do we know about how herbivores can structure seagrass meadows? Well, herbivory can change the community composition in a seagrass meadow. So by creating a disturbance, they can favour recolonization by faster growing seagrass species, which can cause a shift to more of a pioneer community. And this can increase diversity as well, depending on the species mix that you had at the start. They will consume epiphytes that are growing on the seagrass blades. So particularly in the case of those mesograzers that I mentioned before, they're feeding directly on the epiphytes, but also all of the other herbivores will be removing the older seagrass blades as they're feeding. And this can benefit the seagrass because the epiphytes will shade them out, so reduce the amount of light that's reaching the leaves. They are consuming seagrass biomass, obviously. So you can see in this photo the feeding trails left behind after a dugong has been feeding in a seagrass meadow. So they're feeding on both the above and below ground parts of the seagrass plants. And so they can have a big impact on the amount of seagrass biomass in the meadow. Whereas if you think about how a turtle feeds, they'll go along and crop the tops of the blades, which can cause reductions in shoot height. And we also can see reductions in shoot density sometimes. And this can impact the ecosystem services that the seagrass meadow can provide. So for example, if we have intensive green turtle megaherbivore grazing in this part of the meadow, we can see those reductions in structure that I just spoke about. So less biomass, less shoot height, reductions in shoot height and shoot density as well. And this might mean that that area of the seagrass meadow provides ecosystem services in a bit of a different way to the rest of this seagrass meadow that's not being grazed. So the ungrazed meadow has a lot of structure, and so it might be a better habitat for fish and a better nursery ground, so a better fishing ground overall. Because the below ground parts of the plant aren't being disturbed, it will be able to keep carbon that it's stored locked up in its sediments. And it'll be trapping sediment, so might be better at protecting coastlines from flooding. Whereas this grazed part of the meadow will be providing different ecosystem services. So it's acting as a really good food source for the green turtles. And if seagrass productivity increases in response to herbivory, which has been documented in many cases, but not always, then it might take up nutrients more effectively from the water column. And it might also be a really important tourist location because tourists love turtles, right? They're not so keen on just seagrass meadows, but turtles they'll definitely go on a dive for. And so we put all of this information together in a kind of conceptual model, looking at how herbivory might vary in different states of a seagrass herbivore system. So what we've got here is this kind of continuum where you've got a seagrass dominated state at one end with high levels of seagrass structure, lower levels of herbivory. And at the other end, you've got a mega herbivore dominated system with less seagrass structure and more herbivory. And at the extreme end of that, your seagrass meadow might be overgrazed and would collapse. So potentially, if you wanted the delivery of the biggest range of ecosystem services, you might be looking for a balanced system where you've got some seagrass structure and some herbivory, keeping that diversity and productivity up. And that would give you the biggest range of ecosystem services, depending on what your priorities are as a manager. So we're just trying to conceptualize this and think about how the services might vary in these different states of herbivory. But it's important to remember that there are some positive impacts of herbivory as well. So, for example, tagged turtles and dugong have been used to find new seagrass meadows in the Seychelles. So these turtles were tagged in the Indian Ocean, and there were a few of them congregating in this one patch which hadn't been described, or sorry, hadn't been visited before. And then surveys showed that there was indeed a big seagrass meadow there, and that's what they were feeding on. So they can help us to find new seagrass meadows. And they can also spread seagrass seeds. So this is the research of another PhD student in my lab, Sam Toll. And her work looks at the dugong and sea turtle poo. So that's what she's holding in that photo. And she has found that, like the herbivores on land, green turtles and dugong can consume seagrass seeds and then spread them by swimming around and then pooing them out the other end. So they can spread the seeds up to 500 kilometers, and they can help to contribute to the genetic diversity of seagrass meadows, help seagrass meadows to re-establish by spreading these seeds. And so these seagrass meadows are really well adapted to cope with herbivory. They've evolved under really intense herbivory, and the herbivores perhaps have been able to help in maintaining those seagrass meadows too. So it's definitely not all bad and all negative impacts at all. So first up, I'm going to talk you through how I looked at my first aim, which is investigating how the herbivore community as a whole can structure seagrass meadows. So all three of those herbivore groups I introduced you to earlier. So to do this, I carried out an experiment down in Gladstone, which is the southern end of the Great Barrier Reef, a really industrial location in the harbour there, but also really large areas of intertidal seagrass meadows and large populations of particularly megaherbivores, so both green turtles and dugong grazing on the seagrasses here. So I set up this experiment at a meadow called South Trees within Gladstone Harbour, and I had 3 sites which were a couple of 100 metres apart within this meadow. So I used exclusion cages to understand the impact of herbivory. I had different types of exclusion cages to exclude different groups of herbivores. So to exclude the megaherbivores, I had these steel cages, which were 2 meters by 2 meters, half a meter high. To exclude macro herbivores, I had modified crab pots, which were covered in fishing mesh. These would exclude the larger herbivores as well. And then lastly, to exclude the mesograzers, I use carbaryl deterrent blocks, so an insecticide infused into a plaster block, which excludes the amphipods and isopods, particularly just in a small area. And because I wanted to look at the interactions between these groups in structuring the seagrass meadow, I combined some of these treatments to exclude multiple groups. So I had these 3 key treatments that I just described to exclude one group. Well, the middle one excludes 2 groups, but to target these 3 key groups. I had control plots which were pegged out in the meadow. I then had a carbaryl block inside a megaherbivore cage, and that only allowed the macro herbivores to access the seagrass there. And I had a carbaryl block inside a macro herbivore cage, which kept all of the herbivores off the seagrass. I then had 2 procedural controls just to account for the presence of the cage and the plaster block in the meadow. So all of these treatments were left in the seagrass meadow for 3 months. And then at the end of the study, I looked at the differences with the naturally grazed meadow and my exclusion treatments. So like I said, I had 3 sites laid out in the seagrass meadow that were a couple of 100 meters apart. And at each site, I had 3 replicates of each treatment randomly allocated to a spot on the grid. So at the end of 3 months, I took seagrass cores and I took photos throughout the experiment to monitor change. And you can see that after 3 months, within the macro herbivore and mega herbivore cages, there were some obvious differences inside and out just looking at them. And you can see in image D there, we had some dugong feeding trails really near to one of the sites. Unfortunately, not through the middle of the site, but you could see that there was definitely dugong feeding in the area, and we observed lots of green turtles while we were there too. So looking at the data, we can see that this is the change in the above ground biomass between the start and end of the experiment, estimated using photos of all of the plots. There wasn't any difference in the response at each site, so I've combined all of the sites here. And you can see that the plots that were open to mega herbivore grazing didn't change very much throughout the experiment. So a little bit of a change in above ground biomass, which was the growing season, but very small compared to those treatments that excluded the megaherbivores, where we saw big changes in above ground biomass. And same with shoot height. So we see the same pattern where there's less change in those grazed treatments that the turtles could access, but where the turtles couldn't feed, we saw increases in shoot height at the end of the experiment. So essentially what we're seeing here is long seagrass where the turtles couldn't feed, and where they could feed, they were keeping it short across the whole site. Similar pattern with shoot height here. And you can see there's not really any additional impact of excluding the fish over and above what we're seeing with excluding the megaherbivores. So they're the ones that are really driving this change. And the interaction analysis on this data also showed that. In terms of the below ground seagrass biomass, we just looked at cores at the end of the experiment here, and you can see there's no difference between the different treatments. So what we're seeing is turtle and possibly dugong feeding that's impacting the above ground seagrass biomass and cropping it very short, but not impacting the below ground seagrass biomass at Gladstone. So what about the mesoherbivores? Well, we saw reductions in the numbers of mesoherbivores when we had the carbaryl blocks, but they weren't really massive reductions. And what we can see here is that the populations of mesoherbivores were very small. And we also had very low epiphyte loads on the seagrass blades. So although we can't be completely confident that we were excluding all of the mesoherbivores with the carbaryl, we can see that both the numbers of mesoherbivores and epiphyte loads on the seagrass are very small, are very low at Gladstone. So it seems like the mesoherbivore epiphyte interaction isn't very important here, which is interesting when we compare that to other locations, temperate and subtropical, where this interaction has been shown to be really, really important. So the conclusions from this interaction study are that megaherbivore feeding has had the largest impact on above ground seagrass structure. And this impact was seen throughout the whole meadow. Instead of being concentrated in any one area, we were seeing these meadow scale effects. We didn't detect any impact of the other herbivore groups that were present in lower abundances. So there were some herbivorous fish in the area, but not having detectable impacts when you compare that with the big impacts of the turtles. So we didn't detect any interactions between the herbivore groups. It could be that the megaherbivore grazing is keeping this seagrass meadow in a state of lower above ground biomass, which is making it less favorable for the other herbivore groups. Perhaps that's one hypothesis to explain it. So I don't have a link to a publication on this at the moment. It's currently under review, so hopefully soon. So now that we know that the megaherbivores are really important in one location within the Great Barrier Reef, what about a different location? So we wanted to focus in on Megaherbivory in another location. So I carried out a similar experiment in Green Island, just off of the coast of Cairns. So this is one of the oldest marine protected areas on the Great Barrier Reef. And it's surrounded by large areas of a diverse seagrass meadow. And you can see there in that photo just how long and lush and diverse that seagrass is. You can see a few species just there. So this experiment was carried out in the shallow subtidal seagrass at Green Island. There's lots of green turtles around Green Island as well, and it's a really important location in terms of tourism, so quite different to the Gladstone site that I used before. So it was a really similar experiment to what I ran down in Gladstone. I had 3 sites, again, a couple of 100 meters apart. At each site, I had 3 exclusion cages, so looking like this, and I left them in the seagrass meadow again for 3 months. So you can see here just how long and lush the seagrass looks inside and outside of the cages at Green Island. And it pretty much looked like that at all 3 sites for the first 2 months of the study. And after 2 months, we jumped in the water. We'd monitor every 2 weeks. And so we'd go back and we monitored sites 1 and sites 2 in October, and they looked exactly like this. No obvious differences inside and outside of the cages. But we jumped in at site 3 and we noticed that all of the seagrass was pretty much gone. There was a really, really big reduction because before it was looking like that picture of the August when we set up. And so we thought that we'd perhaps observed some grazing event, but we couldn't be quite sure that there wasn't another reason that the seagrass had disappeared in this one area. So we went back and we put a GoPro camera down. I don't know how well this video will work through Zoom, but hopefully you can see that we did catch green turtle feeding around one of our cages. So we knew that this loss in seagrass was due to green turtle herbivory. And it was interesting because we kept monitoring this one site for the next month, and this grazed plot where the seagrass was being eaten by the turtles, it didn't get any bigger really. It was about a 50 by 50 meter area, but it did get a bit barer throughout the rest of the experiment while we were monitoring over the next month. And this is the first time that this kind of grazing plot formation has been observed by turtles in the Great Barrier Reef. So herbivores on land famously create grazing plots, and green turtles in other seagrass meadows throughout the world have been observed to create these grazed plots, graze the nutrient-rich regrowth and kind of move between the plots. But we've never seen this feeding behavior on the Great Barrier Reef, so it's really interesting. And in terms of how this looks in the data, pretty much as you would expect. So we took seagrass cores at the end of the experiment and compared these treatments at all three sites. So you can see in terms of the above ground biomass at sites 1 and 2, there was no difference in the controlled and caged treatments. Whereas at site 3, where we saw the turtles grazing in the controls, above ground biomass was much lower, and the seagrass was still nice and long inside the Megaherbivore cages. And it was a similar pattern in terms of shoot density. These differences weren't significant, but you can see there's a clear pattern towards lower shoot densities in the control plot at site 3 at the end of the experiment. And so perhaps if we kept recording for longer, we might have observed even more changes in shoot density. And if we look at the shoot height data over time, you can see clearly at site 3, when the turtles moved in and they started grazing, there were some big reductions in shoot height inside the controlled treatments, whereas at the other 2 sites, there weren't really any differences inside and outside of those cages. Now, in terms of the below ground seagrass biomass metrics, we saw, again, a similar pattern where we've got long seagrass inside and outside of the cages at sites 1 and 2, and then also inside the Megaherbivore cage at site 3, high below ground biomass. Whereas in that grazed plot, you've seen that we've got the reductions in above ground biomass, but also in below ground biomass too. And this is really interesting because the only time that turtles have been observed consuming the below ground biomass in a seagrass meadow was in this study in the Indo-Pacific in 2014. And you can see that in this study, a lot of the above ground biomass had already been consumed by the green turtles before they started digging for the below ground rhizomes. Whereas in my study, although within that graze patch, the above ground biomass was very low, throughout the rest of the meadow, there was still plenty of seagrass. At the other 2 sites, there were no differences at all. So it's definitely a very different dynamic to this study, but this is the only other time that turtles have been observed consuming those rhizomes and roots. And we did actually catch the turtles on the GoPro pulling out the whole seagrass shoot and consuming those roots and rhizomes as well. So within this grazed plot that was formed at site 3, we had lower above ground biomass, shorter shoots, reductions in shoot density, and lower below ground biomass. So I guess the next obvious question is, well, why were the turtles feeding like this? Could it be that they were targeting a higher biomass area? Well, we've seen from the data that we have that this wasn't a particularly high biomass area and that there was plenty of seagrass biomass throughout the rest of the meadow. So there's probably not that they were targeting this for biomass. Perhaps they were targeting a certain species of seagrass. Well, we have some shoot counts of the plots at every site at the beginning and end of the experiment. Now, there were no differences in treatment, but there was a difference in the community composition at site 3, which was mainly driven by lower abundances of Halodule uninervis and higher of the more colonizing Syringodium. But as you can see, there was still plenty of all of these species throughout the rest of the meadow, so it seems unlikely that they were targeting a certain species. So perhaps they were targeting the nutritional content of that area. And we know that herbivores on land definitely exhibit this kind of behavior. And there have been some studies showing that underwater turtles and other herbivores can show a preference for higher nutrient plant matter as well. So you can see here, we tested the seagrass inside the cages at site 1 and site 3 at the end of the experiment. Now, this was because we didn't have enough seagrass inside those control plots to actually send off and analyze for nutrients, tissue nutrients at site 3 where the grazing happened. So we used the seagrass inside the cages. Now, there could have been some translocation of nutrients through the rhizomes, but this gives you an indication that at site 3, for most of the species that we tested, the nutrients were higher inside at site 3, which is where the turtles were feeding. So it gives us an indication that maybe the turtles were targeting this area based on its higher nutrient content, but it would have been better if we'd had nutrient analysis from the beginning of the experiment. But obviously we didn't anticipate that the turtles were going to form this grazing plot. So it could be that they're targeting the nutritional content of the blades there. But also, as I mentioned, we saw the turtles excavating the rhizomes. And rhizomes are really high in carbohydrates. So perhaps they were feeding in this way to access the rhizomes because they had a dietary need for carbohydrates. So these are all kind of the hypothesis that we're thinking is some kind of a nutritional need for either higher nutrient above ground biomass or accessing those below ground roots and rhizomes. So the conclusions of this study are that green turtles form grazing plots within the Great Barrier Reef, which is the first time we've recorded that, so that's really exciting. And this grazing plot formation has massive impacts on the above ground seagrass structure within that small area, which is very different to what we saw in Gladstone, where the impacts were across the meadow as a whole. We saw below ground biomass being consumed, which is a pretty novel observation as well. And perhaps this feeding is motivated by the nutritional requirements of those green turtles. There's a bit more work to go on to figure that out, I think. So this has been published last week in Marine Environmental Research, if you want to read the paper. And as I mentioned, the ARC project that my PhD is a part of also is looking at Megaherbivory. So we've been looking on a more GBR-wide scale at the impact of herbivory in seagrass meadows using exclusion cages again at all of these sites. So there were 8 locations throughout the Great Barrier Reef where exclusion cages were put out, and at multiple sites within some of these locations too. Now, these cages were deployed for a little bit longer than my cages, so deployed for 12 months to get an idea of the more long-term impacts of herbivory. And in a range of seagrass habitat types throughout the Great Barrier Reef, and these were sampled quarterly. So I'll give you an idea of some of the results that we've had. Again, we're just working through this data, but you can see from some of these images that there's a visible difference inside and outside of the cages. So this is up at Ingram Island on the reef flats. At Lizard Island, just outside the research station, we saw some differences. You can see there inside and outside the cages, but also saw the turtles just lovingly gazing into the cage at the nice long seagrass. And at Green Island, so these cages were put out after I finished my experiment, and they were put really close to where I had my site 2, which was ungrazed. And these cages, again, were completely ungrazed for almost the full year. And then at the end of the year, we saw the turtles initiating another grazed plot in this location, in a really similar location to the first one that we'd seen, but a few hundred meters away. So indicating that this is a repeated behavior and that they're moving between different grazed plots. So again, really interesting. And at Yule Point in the intertidal seagrass meadows up there, we could see a difference inside and outside of the cages. And the field data from the Cymodocea and Thalassia meadows, it's a mixed picture, but we do see some impacts of herbivory on canopy height after a year. So you can see at Lizard Island, One Tree Beach, and in the Green Island Lagoon, there's differences in the cage and control plots. And we had 8 cages at every site here. So But whereas at the Green Island back reef, we didn't see really any impact of the herbivory throughout the year. And in the Halodule meadows, so the lizard research beach where we saw the turtle gazing into the cage, we can see an impact of reduced canopy height in the control plots in blue there. And also at Yule Point, so just north of Cairns, whereas at Bowen, we weren't really seeing any impacts. So we'll be working through this data and looking for patterns. But what we're seeing is that Megaherbivory is structuring these meadows, a lot of these meadows within the Great Barrier Reef. So lastly, I wanted to look at the impact of herbivory by urchins and fish, so the macro herbivores. And again, here I worked at Green Island. So when you're at Green Island, you can see urchins feeding in the seagrass meadow, and you can also see herbivorous fish in the area. So it's an interesting place to look at macro herbivory. And if you're snorkeling around, you can also see lots of bite marks on the seagrass blades. So I had 3 sites here around Green Island to look at the impact of macro herbivory. Sites 1 and 2 were on the intertidal reef flat, and site 3 was in a similar but slightly shallower location to where I had my megaherbivore cages. So to understand the impact of macro herbivory on the seagrass meadow, I used exclusion cages. Again, these were deployed for 6 weeks. I updated my procedural control to have some mesh on, because at Green Island, the cages got very fouled. So I wanted a procedural control that would take that into account. And I went out and I cleaned the I cleaned the exclusion cages twice a week. We did sometimes tell people we were cleaning the coral reef because the tourists thought we were quite insane, but we were just cleaning those cages to make sure that there's plenty of light getting to them. We also did seagrass tethering, so harvested blades of seagrass from the meadow without any bite marks on at the start. We took pictures of those. I spliced them into these ropes. I had 10 ropes per site, and I put them out into the seagrass meadow for 4 days and recorded the number of bite marks and the surface area that was lost to macro herbivory at the end of those four days. I carried this out every 2 months for a year. I also put GoPro cameras down to try and record who the herbivores were. So I'm just working through a lot of these results now, but I used these 2 species that were the most common in the meadow as a whole. And you can see that there was herbivory overall was quite low at all 3 sites on both species, but we did have some variation over space and time at Green Island in the amount of surface area lost to herbivory. And if we look at who the herbivores were, you can see that we had in the blue, dark blue and light blue there, that's large and small fish. We also had herbivory by urchins. You can see the shreds that they leave on the blades, and turtles as well. And that was on both of those species. Again, that's quite variable over space and time. And it's really quite low for much of the year compared to other tropical locations. So here we were seeing that the macro herbivores were consuming between 1 and 46% of daily seagrass productivity, but it was mostly in those very low numbers. Whereas in other tropical locations, we've seen that herbivores, macro herbivores, can consume all of the seagrass productivity and up to 10 times the seagrass productivity, particularly in marine protected areas as well. And so we've seen that this macro herbivory is at quite a low level and it's not having an impact on seagrass meadow structure. So you can see here the cages controls and procedural controls. There weren't any differences in the above ground biomass or in the shoot counts within these cages. So while herbivory is taking place at a low level, it's not impacting the meadow structure. And in terms of trying to catch herbivores on the GoPro, well, I wasn't really very successful. I caught a lot of really cool animals moving through the seagrass meadow, but I didn't manage to find any animals feeding, apart from turtles. So I didn't catch many macro herbivores moving through the meadow. So we've seen that there is herbivory happening at Green Island by small fish, large fish, urchins, and turtles, megaherbivores. It doesn't impact the above ground seagrass meadow structure. And it varies over space and time, and it's at quite a low level. And it's interesting because often in marine protected areas, we'll see really high levels of herbivory as those fish populations increase. But perhaps what we're seeing at Green Island is a marine protected area where the whole food chain is protected. And so the predators of these macro herbivores are also protected, and they're keeping their populations in check, which means that herbivory isn't really exceeding daily seagrass productivity. It's just at a very low level throughout the year. So this is one of the first of these kind of studies that's been done in a highly protected, marine protected area where predators are also present. So really interesting to see the dynamics there compared to what we already know. So in terms of the overall conclusions of my work and feeding into the overall project, if we take it back to that seagrass ecosystem service model that we came up with, we'd probably put Green Island at more of a balanced seagrass dominated end of that spectrum. Whereas down in Gladstone, it's more of a Megaherbivore dominated system, definitely shifted a lot more to that direction. And this is important because the way that we monitor seagrass meadows throughout the Great Barrier Reef is by visually assessing the biomass, so snorkeling or with a helicopter drop-down camera. And so this gives a score based off of the biomass in the meadow, the species that are present, and the area of the meadow. And so if you were looking at this seagrass meadow, you might think that it's in quite a poor condition if the biomass is low and depending on the species makeup of that meadow and the area that it covers. But if you could understand that this was just a meadow where seagrass productivity was being consumed by megaherbivores, then you might be able to change your score and realize that this is a seagrass herbivore system that's perfectly in balance and that it's not actually in a poor condition. And this is important for the way that we monitor seagrass meadows throughout the GBR. And it also can give us an insight into tipping points and perhaps where, if meadows are starting to become overgrazed or there's other issues, if we understand the herbivore system, seagrass herbivore system as a whole, then we get a better idea of the health of the seagrass and the herbivores that rely on it. So in summary, megaherbivores have the biggest impact on Great Barrier Reef's seagrass meadows of all of the herbivores, which they can consume the largest amount. So I guess that's hardly surprising. But the mode of megaherbivory is different between different sites. So we've seen in Gladstone, they can feed throughout the whole meadow and consume the biomass across the site. Whereas at Green Island, they're focusing in and consuming all of the biomass in quite a small area. We've documented new herbivore feeding behavior, so formation of those grazed plots and consuming the Belowground seagrass material. And we found that mesoherbivores are present, but they weren't playing a large role at the site that I looked at. And we've had some more research looking into the role of mesoherbivory in the GBR from one of our honours students who's just published that. And he's found that mesoherbivory can be important. So just at that site that I looked at Gladstone, it wasn't playing a huge role. And macroherbivory can be important in Great Barrier Reef seagrass meadows. It's not a structuring force, it's happening at quite low levels, but perhaps it's being kept in check by a relatively intact food web within that marine protected area. So if anybody has any questions, I'd be happy to take them.
Thanks so much, Abbi. That was really great. And yeah, congratulations on your papers as well.
Thank you.
So if anyone has any questions, if you could pop that in the Q&A below or just make a note of it there and I'll let you ask your question. Abbi, I just wondered, is there a way to exclude dugongs or turtles? Can you separate out those things, or is it not possible?
No, I don't think it would be possible because we've just got those big cages to keep them out. So you can kind of see more evidence of when the dugongs have been there because they create these feeding trails through the meadow. So you can get a bit of an idea of which of your herbivores is causing the disturbances. At Green Island, we rarely see dugongs, so we're pretty sure that that's all due to green turtle herbivory. But yeah, it would be really nice to know which is the culprit for consuming all of the seagrass for sure.
Okay, thanks. You answered my second question there as well, because I was wondering about the dugongs at Green Island. So Ricardo has a few questions for you. I've just unmuted you, Ricardo. You might have to, hang on.
Hi.
Okay, there you go.
Hi, thank you. Thank you. So yeah, I was writing, but I can just, hi, Abby, thank you for your presentation. It was really, really good. So I have a question. One is, have you find any seasonal pattern in grazing? Like if there is a difference between maybe summer, winter, because I know that seagrass change, like they're very dynamic, they change really often. So
Yeah.
I was wondering.
So that's probably something that we'll be looking at as part of the ARC project when we're analysing all of that data throughout the whole Great Barrier Reef, because those cages were out for 12 months. And so you're right, a lot of the seagrass meadows are quite seasonal. They have the growing season in summer. Whereas Green Island, less so. The seagrass meadow there is quite constant. There's some changes in productivity, but not as many. So you've got, yeah, the growing season of the seagrass, but then also when the herbivores are moving in and out. So it's kind of interesting to try and figure out which is the dynamic there that's causing the growth in the seagrass, or is it being more grazers moving in? You've got the movement patterns and the seagrass growth to consider. But that's definitely something we'll be looking into, whether the herbivory is having more of an impact when the seagrasses are senescent, and we'll be able to look at that with the data from the ARC project. I haven't been looking in depth at that because I've been trying to finish off my PhD, but I'm sure that that data will have some answers on seasonality, and that will be really interesting. Yeah.
Okay, no, thank you. Yeah, and only one more, because right now I just started my master, and so I'm looking at World Heritage property that have seagrass meadow within them, and some properties is pretty clear, but some others are a bit difficult because maps are lacking, and also research in some places in the world is really scarce. Yeah. So how much do you think the presence of green tartar is related to presence of seagrass? Is almost certain, or is possible that they just go there, maybe nest and then go away without grazing?
I think that's probably a complicated story to tell, because green turtles can graze on other things and their diet does change throughout their life. And definitely when they're going to nest somewhere, they might not necessarily be feeding. But we've found if you've tagged green turtles, and they tend to be quite location specific to their foraging grounds when they come back as juveniles and start to forage in seagrass meadows. So they don't tend to move very far. So if you were to tag a green turtle and find, or a few green turtles, and find that they were just hanging around in one area, it's likely that they're foraging. Now they could be foraging on seagrasses, or they might be, if they're out on the reef, foraging on algae, on something different. But yeah, if you knew where they were, you'd probably be able to figure out what it was likely that they're grazing on, and if seagrass was a potential candidate. But yeah, they are often grazing on the seagrass meadows there because they're a very good food source. So yeah,
okay,
often, but not always.
Yeah.
Thank you. Are there any other questions for Abby? Okay, Abby, we might leave it there. Thank you so much for your time. That was a really great presentation, really clear, and yeah, I certainly learned a lot. So thank you, and good luck with the rest of your PhD.
Thank you. Thank you very much for having me.
Thank you.